Hello guys, welcome to Big Book. Today we are going to discuss the new topic, estimation of blood glucose by Folin method. And one more update is that we are going to launch a new logo, this one. So the old logo will get decommissioned in a while with the upload of a few more new video logs on different topics as per the request. And now we are going to discuss the experiment. So here this estimation is the quantitative method, where we are going to estimate the amount of the glucose present in the blood. Well guys, in this slide we are going to discuss the principle of the experiment, where the reducing sugar like the glucose is going to reduce the cupric ions present in the alkaline copper reagent to cuprous ions, or to the cupric sulfate is converted into cuprous oxide. So here whatever the copper sulfate is present in the copper reagent is get converted to the cuprous oxide, or the whatever the copper compound is going to present within the copper reagent, that is going to get converted to the cuprous ions. So here the compound used within the copper reagent is the copper sulfate. So here let it be the reference to the copper cuprous ions or the cupric sulfate. So both the ions is the sense within the solution medium, the copper sulfate is going to get breakdown due to the reduction, that is the glucose or the reducing sugars are going to reduce the compound, so cuprous ions forms. So the reaction gives the cupric sulfate. So that is what the compound copper sulfate is going to get reduced to the cupric sulfate of the same form. So that is converted into cuprous oxide. Now here this cuprous oxide is get reduced again by the phosphomolybdic acid. So here this cuprous oxide is going to reduce the phosphomolybdic acid to phosphomolybdous acid. So here, which is then gives the color blue to the whatever the solution is present within the test tubes or the samples. So with the help of this blue color, we are going to measure the optical density within the 420 nanometer, or in some experiments 490 nanometer. In some experiments that is seen to be as the 610 nanometers, like this. It differs with the laboratory. So here we are going to use 420 nanometer as the absorbance optical density within the calorimetry. So this is the reaction. Here you see the reducing sugar reacting with the copper reagent, and this is what the cuprous ions or the cuprous oxide. Further here the next, well guys, in this slide we are going to discuss the reagents required. The first reagent required is alkaline copper reagent. The second reagent is the phosphomolybdic acid reagent, and the third is the standard glucose solution, and fourth is the protein precipitate. Now here alkaline copper reagent is the one where we are going to use it for the principal purpose, and also the phosphomolybdic acid. So here it has been given very clear composition or the ingredients to be used, how and what the weight or the mass we are going to use in what volume of the distilled water. So it is going to be this particular chemical formula. For the phosphomolybdic acid also there is a chemical formula to prepare this particular solution. So all the description which has been given is the correct one. And also standard glucose solution, how it is prepared. And here this particular reagent, I can tell that you can differ this with the requirement as you want to make. So standard glucose solution you can differ, so that you can get the different values or the different readings, or to examine your, or to test your students or the candidates. You can use the different concentration. And the protein precipitant is the one where this is what the last one, where this has to be prepared as exactly it has been given, so you can use it in even in the longer period also, because it is not going to get spoiled, or the formula is not going to get errored. So well, in this slide we are going to discuss about the blood sugar levels of the individuals. So here, if you are going to consider the blood sugar levels within the blood of the individuals, if you are going to consider the individual post-absorptive state is nothing but it is after the overnight sleep, that is after the dinner we are going to sleep very well, and we are going to get up in the early morning. So then we are going to find the sugar level to be as 60 to 110 milligrams per 100 ml. So here, post absorptive state, the blood is going to have a very varied range of 60 to 110 milligrams. So in varied range, in the sense, if you consider 100 ml as the sample of individuals, 100 individuals, if you are going to take a sample, so somebody can have 60 mg of blood sugar level per 100 ml. Some FU, that is going to have like 72 mg, and if you can have 85 mg, and if you can have 100 mg, and if you can have exactly 110 mg also. So this is how the varied differed individual glucose levels within the individuals, that is post absorptive state. And now we are going to consider the blood sugar level is found to be raised by 10-20 milligram in the state of anger, fear, anxiety and pain. So here, during this time, the energy boost is going to happen. So there will be the release of the energy molecules that is stored in the form of the energy storage of the, either in the form of the glycogen or lipid or the carbohydrates which are present, especially lipid and the glycogen are going to get breakdown. So further it is going to give some boost up within the blood sugar levels, that is from 10 to 20 milligrams approximately, during the anger, fear, anxiety and the pain. So here, for a normal human being, after having the food, intake of the food, the carbohydrate digestions, or whatever the protein or whatever the food he has been taken intake, so that is going to raise the blood sugar level from 130 to 140 milligram per 100 ml. So here, immediate or the post having the food, that is the intake of the food, is going to increase the blood sugar level to 130 to 140 milligram per 100 ml. This is very much differed from the diabetic patient. It is not compared with the diabetic patient. Diabetic patients may have more raised sugar levels compared to the healthy normal individual. So next is the after 1 to 3 hours of sugar level falls to normal. It is nothing but if you consider after the intake of the food, if you consider after 3 hours, it's in some individuals, if they are very active and hard workers, there can be fall in the sugar levels within an hour itself. In few individuals who are having a very less hard working activity, there can be this fall of the sugar level even after the 3 hours. So till 3 hours the sugar level can be 130 to 140 milligrams per 100 ml. So here, this difference is because of the breakdown of glucose in glycolysis cycle and also the tricarboxylic acid cycle. These are the two major cycles which are consuming the glucose, where they are going to break the glucose and forms the adenosine triphosphates, that is the ATP molecules, which are required for the further biochemical reactions within the cells. So here, apart from this, the different major other ATP generating cycles which are present or phenomenally happening within the cells is going to make use of the glucose. Because of that, there will be the fall of the glucose level within the blood sugars. So hence, this is the small discussion about how an individual can differ in his blood sugar levels depending upon the situation or the condition. So it is better to have this knowledge before taking the sample into consideration. And to, well guys, in this slide we are going to discuss the procedure. In the procedure, it is first and foremost thing is that we are going to have a blood sample, okay, of 5 ml of the blood sample, where we are going to separate the proteins from the blood sample, because we are going to take the estimation of the glucose present in the blood sample but not of the proteins. So we should have a particular concept of separating the proteins from the blood sample. Hence there should be a protein precipitant, where we have already discussed about the protein precipitant reagent, which is going to help us to separate the protein. So now the procedure is that we are going to pipette out 5 ml of the blood sample into a centrifuge tube, which is already having a 3.5 ml of the distilled water, and further we are going to add protein precipitant of 0.5 ml. Then we are going to swirl or mix, and again we are going then we are going to centrifuge for 5 minutes at 5000 rpm. So highest the rpm, the high molecular weight is going to get precipitated. So here proteins are going to get precipitated, so that precipitate we are going to discard and a supernatant is going to be retained, and further we are going to use that supernatant for the further estimation. So now the actual procedure of the estimation starts. So first we have done the extraction. Now we are going to do the estimation by using the standard glucose solution reagent also, and the whatever the sample blood sample glucose, which we consider it to be as the unknown, that we are going to take into consideration. So here to do this, first we are going to take 6 test tubes. In this one test tube we are going to mark it to be as the blank, and the rest of the 5 test tubes we are going to take the different volume range of the standard glucose solution, that is of 0.2 ml, 0.4 ml, 0.6 ml, 8 ml, 0.8 ml and 1 ml. So this all 6 test tubes has been marked, one as the blank and other as the different volumes of the standard glucose solution, ranging from 0.2 to 1 ml. And other 2 test tubes we are going to consider, where we are going to take 1 ml each of unknown blood supernatant solution. So here, that is whatever the extract we got from the blood in each test tube, in both the test tubes 1 ml we are going to take. Now here we got 6 plus 2, 8 test tubes. Now we are going to make up the volume to 2 ml in all the test tubes. So here whatever the lesser volume is there, that we are going to make up. In the sense, for the blank we are going to add 2 ml. For the rest of the standard different ranges of the standard glucose solution, we are going to add whatever the remaining amount which will make up the test tube volume to 2 ml. That one, that is for 0.2 ml of the test tube, we are going to add 1 ml of the distilled water. Like that, whatever the lesser amount is present in the other test tubes, to that we are going to add according to it. And for the unknowns also, we are going to add 1 ml of the distilled water. So the makeup is going to be 2 ml. Now we are going to add alkaline copper reagent, whatever the copper reagent we have prepared already. For that, we are going to add 2 ml of the alkaline copper reagent in each of the test tube from blank till to the unknown solutions. And further, we are going to mix the content and keep them in a boiling water bath for 10 minutes. So we are going to swirl it or mix it as per the facility present within the lab. Else you can do it in your hands also. You can rigorously swirl, and you are going to keep it within the boiling water bath for 10 minutes. Cool the test tubes and do not shake, and add 2 ml of phosphomolybdic acid reagent, and you are going to wait for 10 minutes and you are going to mix the content. Now you are going to mix the content, and further, according to the presence of the glucose within the those samples, let it be the standard test tubes, let it be the unknown solution, according to that the color change will be observed. So here further we have to do one more thing, that we have to make up the volume to 10 ml. So whatever the volume we have considered, it is up to this, it is 6 ml. So we are going to add 4 ml of the distilled water to make up it to 10 ml within the test tube. So after making up it to the 10 ml, we are going to observe. Well guys, this is the slide of the protocol. The protocol is as exact what we have discussed in the procedure. That is, the first column is of the 8 test tube serials, next column is of the volume of glucose in ml, and next is the amount of glucose in micrograms, and next is the amount of distilled water in ml, which we are going to make up to the 2 ml. Next is adding the 2 ml of alkaline copper reagent to each test tube. Then is mixing the content and keeping them in a boiling water bath for 10 minutes. Further is the cooling under running water, and we are going to add the phosphomolybdic acid, and further we are going to pick up the volume to 10 ml with distilled water and observing under the optical density OD of the 420 nanometer against the blank within the calorimetry. So this is the protocol. Well, in this slide you can observe that we have considered the 2 columns from the protocol, that is the amount of the glucose in micrograms and absorbance at 490 nanometer. So here absorbance at 490 nanometer, we got these readings, and we are going to plot these readings against this mass of the standard glucose solution. So further we are going to make a what we call pass through, or the pass through within this particular readings of the unknown solution, so we can get a exact or the approximate amount of the glucose present within the blood sugar. So here the graph what we have got is amount of glucose in micrograms. This is micrograms, that is 40, 80, 120, 160 and 200, and this here we have made the range of the absorbance, that is 0.1, 0.2, 0.3, 0.4, 0.5, 0.6, 0.7. The units on the x-axis is 1 centimeter is equal to 1 unit, which is equal to 40 microgram of the glucose. So other is the 80, 120, 160, 200. Here it is absorbance is similar, y-axis 1 unit is equal to 1 centimeter is equal to 1 unit, further is equal to 0.1 absorbance reading, 0.2, 0.3, 0.4, 0.5. So here this curve shows the absorbance at 490 nanometer, these readings. So here which are correspondingly for the mass of the glucose. So you can see here, 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 here. So here you can see mass of the glucose against the absorbance against the mass of the glucose has been plotted in the graph. Now these unknowns are having 0.34 as the absorbance reading at 420 nanometer in the calorimetry against the blank. So here you are going to mark them. Here it is going to create an intersection of 0.34 here, and it is going to fall the intersection at this particular point. So it gives 116 as the reading between the 80 and 120. Now here one thing I have to make it very clear that we see that 40, 80, 120 they have been mentioned in between the unit. Here the unit completes at this particular small intersection. Here the unit completes for 80, here the unit for 40, here the unit of 120, here the unit of 160, here 200 micrograms. So here if you are going to consider from here to here 80 to 120 micrograms. So here the particular intersection falls at 116 micrograms. So as this is the slide prepared by the graph plotted by the computer, we are seeing slight difference in its display. So here we can when we are going to plot the same values within the graph sheet, definitely and obviously it is going to give the 116 as the microgram or the reading. So here 116 is the value. Amount of sugar in 1 ml unknown solution is equal to 116 microgram. Okay, that is 116 into 1 ml, that is cubic centimeter. That is 10 to the power of 3 is equal to 116,000 microgram. It equals to 116 milligrams of glucose. So here therefore 100 ml of unknown sample contains about 116 milligrams of the glucose. Here by typo mistake we have printed to 116,100 is equal to 116. So I apologize for the same. So it is directly 116 milligrams of glucose per 100 ml. So this is the experiment which we have conducted in our labs and we got the results. So I hope I am audible and clear for you. If you have any doubts you can ask me in the below comments, and also you can appreciate me and encourage to give new topics as the video logs for at your services, and I will be very much happy to serve you guys. Furthermore, that you also can grow and I also can grow, and I request you all to spread this video log among your friends and other students, juniors or seniors, so that I can get a feedback, let it be positive or negative, and I can give you a better tutorial in the form of these video logs. I hope this is going to help you a lot, and be tuned to Big Book.